Hello students, today I welcome you on behalf of Rasa International School. Today we will do an activity in which we are able to identify acids and bases with the help of indicators. So to understand first how we are able to identify the sample which is being provided to us is an acid or a base with the help of indicator. So indicator is the important part. So what is indicator first? Indicators are the substances which show a change in their color or smell or odor when they brought in contact with acids and bases. इंडिकेटर वो सब्सटेंस हैं जो अपनी कलर को या फिर अपनी स्मेल को चेंज करेंगे जब वो एसिड या बेस के कांटेक्ट में आएंगे. सो इट इज़ वेरी इंपॉर्टेंट. Why? Because without this we are not able to identify the or we cannot differentiate between an acid or a base. Okay, so examples of indicators are litmus paper, turmeric, and many more we will discuss in our further slides. So here we have shown two indicators. One is the litmus paper, which is blue in color as well as red in color, and turmeric, which is in yellow in color. In Hindi we call it as haldi. Okay, so now we are moving on to the types of indicators. So basically we discuss about three types of indicators. First is natural indicator, second is synthetic or man-made indicator, and third one is olfactory indicator. Okay, so as the name is being suggested over here, natural indicator, that means it is derived from nature, and from nature where it is being derived from plants. Okay, so the indicators that are obtained from plants are natural indicators. So examples of natural indicators are litmus paper, okay, red cabbage, turmeric, China rose, and grape juice. So these are the examples of natural indicators. Many more are there. Okay, so these we study today. So in the table which has been listed below, we have three columns. In first column we have name of the indicator and its color, what it color along with acid in the second column, and what it color along with base in the third column. So let us start with blue litmus, that is which is blue in color. So when it is in contact with an acid, it get change its color to red, and when get in contact with a base, there is no change of color, that means it remain as blue. Okay, now red litmus is red in color. Okay, and when it get contact with an acid, there is no change in color, it remain red, and when it get contact with a base, it color change to blue. The third one is red cabbage, which is purple in color, but in contact with acid it turns to red, and in contact with base it turns to green. Okay, so the fourth one is turmeric, which is yellow in color, and when it is contact with acid it color changes to yellow, means remain the same, but when it contact with base it color changes to reddish brown. So here we are able to discuss four natural indicators whose colors are being listed below along with acid and with base. So now we are moving to second type of indicator, which is synthetic indicator. So synthetic indicator is also known as man-made indicator because they are the chemical substances and been prepared in labs, or you can say in industries. So these are the indicators which are man-made chemical substances. Example, methyl orange and phenolphthalein. So basically we have three columns in the table: the name of the indicator, its color, the change in the color when it get in touch with acid, and when it got touch with base. So methyl orange is orange in color, and when it get in contact with acid its color changes to red, and along with base its color changes to yellow. Phenolphthalein is colorless, and along with acid its colors remain colorless. Okay, so there is no change. but along with the base its color changes to pink. Okay, so these are the indicators which are synthetic indicators. Now we are moving to the third category of indicator, that is olfactory indicator. So olfactory indicators are those which change their smell in acids or bases solution are called olfactory indicators. Example: onion, olive oil, vanilla extract. So here we have three columns: name of the indicator, the change of the order when they are get in contact with acid, or change of their order when they are get in contact with base. First is onion extract. In onion extract, there is, when it get in touch with acid, there is no change in smell, means the smell remains the same, but in contact with the base they loses its smell, or the smell gets suppressed, means the smell get not been able to identifiable. Okay, now olive oil or long ka tel. Okay, so there is no change in smell when in contact with acid, but along with the base again there's a loss of smell. Okay, so it loses its smell, or again the smell gets suppressed. Okay, same with the vanilla extract, means for olfactory indicators they don't change their odor or smell along with the acid, but when they get in touch with the base their smell is being suppressed, or they loses its smell. So this is what about olfactory indicators. Thank you, and admissions are open in Rasa International School. You can contact on the mentioned numbers on the given slide. Thank you once again.